All right, conduction velocity refers to how quickly the action potential travels down an axon. It's always going to stay the same speed, right? The velocity is always going to be the same on a given neuron. But some neurons conduct their impulses faster than other neurons. They're always all or nothing. They're always the same speed on the same neuron, but some neurons are just faster than others at propagating their action potentials. And it has to do with how thick or the diameter of the axon, right? That has an effect on the speed, and also how much myelin is on the axon. So we're going to look at both. I think we go through the myelination first. So we'll look at degree of myelination and how that affects conduction velocity, and then we'll look at axon diameter and how it affects conduction velocity. We mentioned in a previous slide a while back that myelin increases the speed of an action potential. So axons that are myelinated conduct impulses faster than axons that are unmyelinated. And myelinated axons go through this process called saltatory conduction, which is about 30 times faster, much faster than if there was no myelin there. Saltatory has nothing to do with salt. Salt means to jump, saltatory, and that's descriptive on how the action potential travels. So to explain how myelinated axons conduct impulses faster than unmyelinated areas, we're going to go to the picture on the next slide. So let's take these three events here one by one. So we're going to start with this first row here. So we're looking at a neuron, and very specifically, we're looking at a dendrite of a neuron. Now we know from previous slides that we cannot have an action potential on a dendrite or on a cell body. Action potentials only occur on the axon, right, from the axon hillock all the way to the axon terminal. So on the dendrites or the cell body, in this case we're on a dendrite, we're dealing with graded potentials, and we know graded potentials die off or get weaker as they travel along the dendrite and cell body. So let's look at the graded potential. So here's the stimulus, and the height of the white area is the strength of the stimulus. And we can see as we travel down this dendrite right here, the stimulus strength, the voltage, starts to decrease. You see the white line or the thickness getting smaller and smaller until we have basically nothing. So graded potential, yeah, strong. In this case, it's depolarized or strong where it first occurs, but as you travel along the neuron, it gets weaker and weaker and just dissipates to nothing. That's a graded potential. Well, we know action potentials occur on the axon, beginning at the axon hillock and moving to the axon terminal. So we're going to go to this picture now, where we take a piece of axon, but if we look at this axon, there's no myelin on it. It's unmyelinated or non-myelinated, you can say either way, compared to this one, which has myelin sheaths on it. So let's start with the unmyelinated axon and looking at a little tiny piece of unmyelinated axon. So there's the stimulus. Look at that little white area is high, it's strong, and it actually starts getting weaker, but then the next channel opens, sodium rushes in, potassium rushes out, right? So you get that that strength of voltage again, just like you had at this channel. And just as it starts to decrease, the next channel opens and you get more depolarization, right? Then the next channel. So all these voltage gated channels, as the voltage is traveling, is opening up the sodium channels to restore the strength of the stimulus at each channel. So even though you get a tiny bit decrease between channels, it restores itself as every channel opens all along this axon, all the way to the axon terminal. So that's the way we preserve the strength of an action potential, and that's how it stays the same from axon hillock all the way to axon terminal, so that it's all or nothing, strength stays the same. Yes, so in this axon you have this velocity of an unmyelinated axon. So what's the difference between what happens here on an unmyelinated or non-myelinated axon compared to what happens here on an axon with myelin. So let's take this axon that has Schwann cells or myelin sheaths. So there's one myelin sheath or one Schwann cell. There's another one. You might remember there's gaps between Schwann cells or gaps between myelin. We call them nodes of Ranvier, or you can call them myelin sheath gaps. That makes sense as well. And what you'll notice here is you have a channel, voltage-gated channel, at this node, but you have no channels, no voltage-gated channels where the Schwann cells or where the myelin sheaths are. Only in those nodes or gaps do we have channels again. So therefore, this channel opens, you get depolarization. You see the white area, how tall it is, so it's strong. But notice how it's getting weaker, but it takes a long time to get weaker. That's because it's insulated. This myelin sheath is insulating that electricity. There's no insulation here, so it starts to drop a lot quicker. So you need channels real close together. So here, the charge is preserved mostly. It's weakening a little bit, but it's mostly preserved along inside that Schwann cell, right, inside the axon. Once we get to the node, well, these channels open up, sodium rushes in, and you restore that previous strength. And going through the next area where there's a Schwann cell wrapping around the axon, the exact same thing happens. So the point here is that we don't have to open as many channels because we're preserving or insulating the charge with the myelin sheath. So channels only have to be open, these sodium channels, at the gaps, at those nodes. So this charge can skip from node to node to node, which is much quicker than having to go through all these channels, many more channels, right, in sequence. So that's why we call this saltatory conduction, because it leaps or jumps from node to node, which is a much quicker way to preserve this charge and an action potential with this myelin as it compared to as if it's unmyelinated. Remember, we said it's 30 times faster than this. So that's how myelin increases the velocity of an action potential and why it's faster than an unmyelinated axon. It was also mentioned that the diameter of the axon also has an impact on conduction velocity. So we're just grouping our neurons or axons into A, B, and C. Group A fibers have large diameter axons, where group C have the smallest diameter axons, and of course B is in the middle with intermediate, and we can see how fast the action potentials travel, meters per second, 150 meters per second, way faster than one. And the difference is, well, it's a much larger diameter axon, and group A also happens to be myelinated, versus group C, the smallest diameter and unmyelinated, much slower than the group A fibers. So those are the two factors, diameter and myelination, to affect the conduction velocity of the action potentials.